But welcome, everyone. Thank you for joining us today for our Innovation Grand Rounds. I'm Stephen Bopart. For those of you I haven't met yet, I'm the Executive Associate Dean and Chief Diversity Officer for Carle, Illinois College of Medicine. And it's truly a pleasure to have as our guest speaker today Colleen Bushell. She is the Director of Healthcare Innovation at the National Center for Supercomputing Applications here. And really, as someone who has dedicated her life to looking at ways of visualizing data for scientific visualization, for information, has been involved in web development from the very start. And I think that's something she's going to touch on today as well. What we want to share and learn about too is the type of innovation that's occurring locally here on our campus and at National Center for Supercomputing Applications. Really, this innovation around technologies that can be applied to medicine and healthcare and biomedical data, and that's why I thought that it would be just ideal to have Colleen speak today about the work that she's been doing. As well as give a historical perspective of many of her experiences, being associate professor of graphic design, where she taught information design. She co-founded a spin-off company called Riverglass that was creating software tools for text analysis and really looking at how data can be meaningful for workflow and to people. So with that, we're very excited to have you here and speak. And she has her presentation loaded here, and I will turn that over to Colleen. But right before, just as a reminder, if you have questions along the way, Colleen had asked if we could just add those to the chat. And then once we get to the question and answer session, I'll be able to pull those out, and we'll pull those from the chat as well as open it up for others to ask questions. So thank you all again for joining us. Thank you, Colleen, and we'll turn it over to you.
Great. Thank you for the introduction and inviting me to take part in your Grand Challenge Grand Round series. My background is different from a lot of people on this Zoom call. My formal education, as Steve said, is not in biology or computer science, but it's in graphic design. So I appreciate the opportunity to share my perspective. I'll talk with you about information design, which is the organization and representation of complex data and how it's one piece of a puzzle to effectively leverage data. And I'll also discuss the concept of innovation and collaboration in my experiences over the years. As you're all aware, there's continual growth of data in the health field, and I wanted to provide a few numbers here to support that understanding. And this shows just a slice of what is produced. And of course, there are other types of data that are relevant to healthcare. And this last year's expansion of telemedicine has really taken off due to changes in licensing and insurance policies to accommodate the pandemic. And this provides yet another opportunity where I'm sure there will be more data generated and more data for analysis. And so improving the use of data can have a great impact on all of these areas that I've listed here. With most of these leading to the improved patient outcomes, which of course is the main goal. There are challenges to finding data that you want, and also being able to bring the right data together to derive actionable information or to support decision-making when you need it. Even when you have information at your fingertips, there are challenges to interpreting the data or the results of analysis. And finally, even if you have the appropriate evidence and interpretation, it's often difficult to communicate that complexity to colleagues or to patients to arrive at confident decisions. So NCSA is an institute on campus, if you're not familiar with it. It's been here for over 30 years, and I've been there at some level since the beginning. And I would say that in general, NCSA's role has been to continually improve the ability to leverage data, whether through advances in computation, cyber infrastructure, analysis, software tools, and technologies that address these barriers. And the role of information design and user experience design has always been part of that mix, but sometimes not as obvious when we talk about innovation. So I realize this is not an audience probably of designers, but I believe it's valuable to be aware of this topic so that you can either engage with designers effectively to address barriers that you face. Or if you're in a position of influence, you can help to facilitate some of these engagements. So I'm going to show some examples of how the design work I've done in the past has been focused on some of these challenges and how it can be applied to current efforts in the health domain. So I graduated in graphic design just as computers were being introduced to the field, and I started working for the newly formed NCSA Visualization Group in 1986. And as an information designer, I spent the early part of my career applying knowledge of visual communication to the design of animated visualizations of data from simulation models. I explored visual treatments to help study and communicate. Here's an example of a severe storm model. One feature of the data was the water density value at each point in a three-dimensional grid. So here we mapped a solid surface, a visually solid surface, to the threshold that radar typically sees. And then we used a more translucent mapping to the threshold of vapor that a human usually sees to provide context for the cloud structure as it evolved through the simulation. I was planning to show some of the animation, but I was very afraid on Zoom that it would really goof things up. So I just have still images for us today. Then we continued to replay the same animation, but this time focusing on different aspects of the data. Here we include the translucent mapping of the cloud structure again for subtle reference and focus on the dynamics of the air movement through the cloud. We also had the wind direction and speed data at each point in the grid. So we developed a few techniques, and one of these is to replicate balls blowing through the cloud by mapping a sphere at selected locations and then following where that would be at the next step. We developed the ribbons by keeping the information about where a point was in the previous few steps. So it became kind of like a timeline for tracing where that point moved. It really helped to understand the airflow better. We even found that using simple visual ideas sometimes provided insight. Here we colored the balls orange when they were rising and blue when they were sinking. And of course, that's not necessary when it's obvious when you're seeing the upward movement in the cloud. But what it did show were wave patterns on the ground and also at the top of the cloud that you wouldn't have noticed if you just used movement. So those are some of the visual techniques we explored and then used over time in other types of visualizations. So exploring new ways to represent data in teaching as an associate professor in graphic design really helped me to refine my approach to design and to methodology. I found that not only did I need to teach students about the formal aspects of visual design, but I also had to teach them about how to continually be creative, how to always explore new ideas, and how to take risks. I found that a lot of people are just uncomfortable spending time on something that's not a sure thing. And it often takes practice to be able to take risks. I don't teach anymore, but years ago, I started a program for undergrads at NCSA that's still in operation. It's called SPIN, which is Students Pushing Innovation. And the goal is to provide opportunities for students to develop their own projects with NCSA mentors and to practice taking chances. This was actually the application form that we used in the beginning. And then now it's still going on, but there's more of a traditional application process that students can apply to. And so if you do know of any undergraduates that are interested, it's definitely a great opportunity to let them know about. After many years of visualizing simulation data, I shifted to software development, eventually incorporating data representation into the user interface. I'll go into more depth about some of these examples, but first I wanted to explain just a few key concepts of information design. So one of the objectives of information design is to provide both a qualitative overview and a detailed quantitative view of the data. So everyone's familiar with how cartography achieves this goal. The qualitative, or the high level view, gives the reader a quick grasp of the content and the relationship between all the parts. And then detail is provided within the same view so that the connection between the qualitative and the quantitative is clear. So often with poor design, only one of these is addressed. Either it's all detail and no high level view to provide context, or it's only a high-level view with no ability to drill down into details. The information designer, in close collaboration with the expert in the particular field, must first determine the hierarchy of information. So I describe this process as kind of determining the series of questions the researcher, the analyst, is asking of the data. Then this hierarchy must be reflected in the form, the visual form, whether it's an interactive flow or a composition of elements in a static view. So a designer uses techniques then for visually grouping and layering information to create this hierarchy. These examples show you on the top what content looks like when it's displayed without visual hierarchy, compared to a design where the important information is made visually stronger than the supportive information. Here's another example of visual hierarchy. This is a project from our team where Lisa Gatsky was the designer. The maps on the left were used prior to her design. And as you can see, her map on the right, in her map on the right, she focuses on the relevant information that the researcher needs to know, and then visually layers the supportive and primary data. Often people will ask our team if we can redesign their existing interfaces to basically look good. And as information designers, we're not trying to make the interface look good. We're trying to make the information clear. And what happens then is people find that clarity pleasing and describe it as looking good. So it's really, we see it as a byproduct of good information design. This is a design not created by our group, but I thought it was a good example to talk about an example in the medical domain. It demonstrates the ability to show a lot of information at a high level about, in this case, a single hospital patient. It uses an approach called small multiples, where each square module is a different test result. And then within each module, as you see in the detailed view on the right, it also shows the compression of time. In the far left column, it's representing a summary of data in the year before admission. Then in the next column, a summary of the week before admission. And then in the larger area, the data shown for each day of that week. Providing this type of overview helps the clinician more easily scan the large quantity of data to determine where there might be areas of concern that then they can investigate more fully. This is an example that I've always liked that illustrates sometimes how a visual representation can provide a different perspective compared to looking at numbers. This is from an article about the fastest runners of the 100-meter sprint and how much faster Usain Bolt was compared to the next best runner. And for someone that doesn't run, such as me, learning that Bolt was 0.11 seconds faster might not seem that impressive. But the viewer can grasp it better when it's mapped to the distance, as in this graphic. It gives a different perspective and relates to something they're familiar with. So one rule of thumb I have is to always provide multiple representations of the same data. This image is from a project we did probably over 25 years ago, and I included it though because it describes one very important task that's always been part of my design process since that experience. And that is to work hard at avoiding misrepresentation. So this is one of our earlier visualization models of that same storm data. And when we created it, we wanted to put kind of a ground plane under the storm to help show the movement of the cloud as it evolved. So we took an image of this land and mapped it on the bottom. But unfortunately, this communicated to other researchers that the model takes into account this particular terrain, which was not the case. So from this experience, I learned that there needs to be a constant checking on potential interpretations of the data. Sometimes the expert does not catch this because their knowledge is so solid that they don't realize how someone less knowledgeable might misperceive the information. There are several ways we try to address this problem by constantly asking the expert to evaluate how could it be misrepresented, by testing examples with various people and reviewing what their understanding, what their takeaway was, and by providing multiple ways to look at the same data to reduce the potential for misrepresentation. And this is part of our approach, whether we're working on medical reports or software. I collaborated on a project with Edward Tufte, who was at Yale at the time. He was a professor of statistics, and he consulted for the Washington Post by evaluating their political poll data. And he discovered that there was always serious review of the text that was published, but not of the graphics that were made to accompany stories. And often those graphics could be misleading. This set him off on a path to improve the design of information. So we worked together on some additional ideas for representing the storm data to incorporate techniques that provided more context and also to add more quantitative information for reference. There's a case study of this project in one of his books shown here if you're interested in learning more. But what this experience definitely made clear in my mind was the need to provide the right amount of evidence for accurate interpretation. And you will see at a later example of a genetic report how we incorporate the evidence needed by the medical geneticist. After this time, I moved towards software development to apply some of these approaches to the design of user interfaces. One of the first tools I worked on was Mosaic, which was the first web browser developed at NCSA. The goal of Mosaic was to combine multiple methods for accessing content at that time on the internet, such as FTP, Waze, and HTTP, different types of servers of information. This is why we called it Mosaic. It was a mosaic of disparate methods brought together into one tool. And it was intended to help scientists share information more easily. You can see in this old picture how we initially had panes, window panes at the bottom of the window to include data with the text documents above. As you can see, the interface is not elaborate visually. We were definitely limited to available widgets, but there were still opportunities to have visual design and user experience design play a role in the development of this new technology. For example, one decision was whether to have the concept of going to content or fetching the content and bringing it to the user. So we had to figure out how to word the phrase in the menu. So technically both occur, but since the content was only temporarily downloaded, we went with the concept of going or visiting a site. Also, we knew we didn't have time to build an HTML editing tool, and we were concerned that people would not go through the trouble to learn how to mark up text. So we included the feature called View Source, which let users display the markup text and learn by example, which I think really did help make the web become more popular. This mosaic icon was a place where I integrated some of my previous animation experience. The browser quickly became popular, and leadership at NCSA wanted me to include the NCSA logo on the interface. And I was adamant that that would waste valuable space. You know, back then monitors were definitely smaller as well. So the compromise I came up with was to have an animated icon to address one of our design challenges, which was how to indicate to the user when content was still being transferred. So this icon is really just, there was an NCSA logo at the time that was similar to this, and we just changed it to include the globe. So using this animated logo for this ended up to be a common feature in other browsers in the early days. So as long as the icon carried meaning, I felt that it was okay to include it. As some of you who may have created web pages early on, you may remember that all the content was flush left or centered, as you see in this early example. And for a designer to not have control over the composition was very frustrating, and I needed better ways to create the visual hierarchy that I described before. So that first year, I taught what was probably the first web design class to graphic designers. And Eric Abina, who is one of the original developers of Mosaic that I worked with, was implemented for me the tables tag, the HTML tables tag, so that I could actually begin to compose the page, the web page. We were able to use invisible images or make the table lines disappear to give the illusion that you could set different objects and text in certain locations. So soon after Mosaic took off, most of the content on the web was more equivalent to kind of like a brochure online. And so the next period, I focused on developing more innovative concepts for how the web could be used. One of these included a virtual office space that we developed for Al Gore's team, which had document sharing in it and discussion threads about policy issues, and a virtual water cooler, which was an informal chat space, which I know now sounds very lame, but at the time those were all kind of new things to try out and to try this idea of making it a collaborative space. So, and I tried to incorporate ideas such as the visual map of the virtual space to give the user an overview of what's there, where they could then click on that visual map and go to a site. We ended up making instances of this for Jim Edgar, who was the governor of Illinois at the time, as well as many of our industrial partners. Another example was to explore the idea of online textbooks. So we made this prototype for McGraw-Hill publisher so that we could kind of show or explore the potential for this form. And so we demonstrated how flexible the organization could be, how to incorporate multimedia as part of the education process. And we explored visual ways to represent the content and to support navigation. We did this with some faculty on campus here in human development. And I wanted to point out that this is another important use of design that we've found, and that's to work through a lot of details and create mock-ups that capture the vision of where things could be eventually. And this helps everyone kind of get in sync with what is needed to achieve that vision. It's something that we still do today in the medical domain. We develop prototypes to help push ideas forward, but then also to refine a lot of those details, because it's easy to come up with really hypothetical ideas, but when you get down to the details, you really start to resolve and develop things that solve the problem. So we take it to be as real as possible so that we can work through all of those issues. And then it becomes more of a kind of a practical roadmap of how to achieve that goal. And then it also creates kind of buy-in for all the stakeholders because everybody's participating. So artificial intelligence is a term that has become popular again, but back in the 90s here at NCSA, there was a lot of work in AI, but we called it data mining and machine learning. And there was a newly formed automated learning group led by Michael Welge. And so I began to work with that team and started to combine my interests in web design that I had experienced with my previous work in data visualization. It really started to develop more user interfaces for this type of data analysis. We did just a little bit of work in the biology domain. Most of it was in other sciences. And this is an image of a tool that we developed for Harris Lewin, who at that time was the director of IGB. And he used it to compare genomes of different species. And you see that interesting picture where we have at NCSA what we call the display wall. And he used to like to put it up on that display wall when he gave lectures so that he could really point to a lot of different detail. Eventually, we co-launched, several of us from the Automated Learning Group co-launched a spin-off company from NCSA called Riverglass. And it focused on software for text and data analysis. Some of the techniques we developed there would be very beneficial in the healthcare space today. Especially one of the things we did was develop methods for making it very easy to query multiple databases all at the same time, and then extract content from those results and put it into a structure that would then make sense, bringing all the pieces together for the person that was doing the analysis. So we included text analysis in that case too, where we could, from structured text, find keywords, of course, but then also from unstructured text, be able to identify certain topics that we knew were relevant and then pulled them out and made them available in this tool. We were able to build what we called link charts and other methods to then work with those pieces of information, always knowing, you know, where the resource was or where the source was that those pieces came from. So during that time at Riverglass, we also worked with some US government entities, including Northcom and Southcom. If you're not familiar with those are two entities that are responsible for managing disasters when they occur. And so I learned a lot working with them about what they called situational awareness and how access and display of relevant data, especially timely, real-time relevant data, is critical. And there are lots of tools that they had that they've developed, and even entire rooms built to manage the display of this type of information. And so obviously I feel like there are lots of similarities there of what's been done there and what's needed in healthcare. And not only for emergency types of situations, but also on a smaller scale and for day-to-day needs. The idea of more easily being able to get a sense of a situation and find the relevant detail that you need quickly. So after about 7 years at Riverglass, I came back to NCSA to start the visual analytics group. And I wanted to apply everything I'd learned up until that point to the healthcare domain. And so this group is comprised of primarily 3 different types of skills: information design, data science, and software engineering. And we work very closely with many different domain experts. We do a lot of work with Mayo Clinic, as well as NIH researchers and faculty here and across the country. And our activities span from developing software tools for research, but also tools used in the clinic, and then even the design of patient reports, for example, for clinicians. So I'd like to go into some detail about a previous project we did with some colleagues at Mayo Clinic, where we applied these techniques to a genetic panel report for hereditary colorectal cancer. It's not as complex as our research tools, but I thought it'd be easier to explain the approach using this example, and then you can see how we use them in more complex environments. And so the audience for this report is not the patient, it's the general clinician or the medical geneticist or a genetic counselor. So of course, genetic information is complex, but the structure of a standard report adds to the difficulty. Usually medical reports seem to be presented using a narrative, kind of a chronological structure. It's as though the person from the lab is talking to the reader, explaining what they did, what they found out, and what it means. So this method of communicating definitely ensures clarity, but it's not conducive to some of the other communication objectives and tasks, such as the ability to grasp the essence of the results so that the details have more context, or to skim the report or to review or refresh your memory. So instead of relying solely on a narrative format, we proposed a structure that is based on the questions asked of the data. So after much discussion with many people, we identified these as the primary or initial questions clinicians want to know. The answers to these primary questions can usually be provided in just a few words. And these answers are available in the report, but it's just that the structure requires a lot of extra effort for the reader to parse out those answers quickly. Each level of these initial questions then has a series of follow-on questions depending on the knowledge level of the reader and the nature of the results. So I described the answers to the initial questions as kind of the summary layers, something that can be grasped quickly so that all the additional detail relates to those main points. And then I call the second layer in the hierarchy the explanation. So this explains the summary in more detail and uses a narrative format there for clarity. And the explanations, though, are organized based on the summary, not based chronologically. So it's easier to find the explanations you need. And then the third layer I call the reference layer. And this is the evidence there that supports the explanations. And then the last layer really just supports questions that cannot be answered in the context of the report, but it's still desirable to direct the reader to the outside resources for further information. So here's a view of the summary layer of the report that we designed. The interactive version is on the left and the printable PDF is on the right. And I'm just gonna step you through just some of this to point out how we tried to achieve some of that. So the most dominant element in the summary view answers the two primary questions quickly at a glance. Is there a problem? and how confident is the lab? Like, what is the degree of evidence for this variation or this variant? So you see also that we have kind of a white box showing the name of the gene, but this is not as visually dominant. This supports the knowledge level of the expert that would know simply by seeing APC what the disorder is and potential actions. It is not necessary then for them to read the brief summary statements to the right. They know they can skip that because they're knowledgeable of that gene. But for the non-expert, we include a phrase such as mismatch repair that describes at a very high, simple level what the gene is involved in, just so that they have some concept of what went wrong. And it's always important when having a set of interpretations that you provide a scale for reference so that the reader understands the entire range or what the options for interpretation were. And then someone that is not an expert may not gain much meaning from that gene name as I described, but instead they will review the content to the right. And this is an example of the interpretation statement. And then here we list the phenotype or the disorder associated with the gene that is relevant to that particular panel. And then here's a statement for possible actions. Here we just put two simple ones, but you can imagine depending on the situation, it may be options for various targeted treatments. And then lastly, in the summary view, we also displayed the remaining genes in the panel and the associated relevant disorders for those genes, which now have a decreased likelihood of being present. And now this page, this is the second page in the report. And here you see, we call this the explanation layer. This layer answers those next set of questions asked by the reader. So it has a short paragraph that clarifies the summary statements and explains really the rationale for the interpretation. There's also brief information about the possible actions that can be considered and any relevant risk information. And then some examples of what I would consider the reference layer. Here, this reference layer provides answers to in-depth questions usually only asked by an expert. So here, information that is specific to the test process is included, and it's usually in that narrative report. But if you're not familiar with genetic testing, it isn't very helpful. But this content is, so it's generally only meaningful for someone that really has extensive knowledge of genetics. But by abandoning the narrative format for this information and putting it in a chart form, it's really easy for someone to quickly find, skim for the information they need, or to completely ignore it easily. And we also felt it was important for clarity to indicate the potential techniques that were not used for this specific test. We also show any literary references that were used for the interpretation. And then interestingly, when we worked with our Mayo colleagues, at the lab, they also learned after we talked with many medical geneticists that the medical geneticists will almost always double check the interpretation that the lab made and do their own analysis of the variant that was found. So we decided to try to help facilitate what they're doing by providing the needed information that you see here pulled out for them to do their own analysis. And then anybody that does not understand genetic information would just pass over this because it's not relevant to them. But these are the key pieces of information that the medical geneticist needs. And in the past, they didn't have those in the report. So often they would call the lab to ask for more detail and then execute their own analysis. And then in the interactive version, we have additional reference information that's not practical to include in a printed report. So here we provide brief material describing the disorder and the gene and the links to the external resources to support any need for more detail. And then in the printed version, we list the sites as well as PubMed IDs for further study. And here I'm showing that we also explored some ideas for helping the expert, like the medical geneticist, assess the interpretation and the relevant evidence that was used by the lab. So here we're displaying it more as a map of the decision process and then which route the lab ended up taking. but then also marking which evidence was applicable to that decision. And then also we put links there to what publications they could access that support that decision. So that project was a prototype. It was actually not ever completely implemented, but because they were just not ready to implement something like that. But that was years ago. And now we're working with them on actually implementing many different types of reports. There's a microbiome report. And then on the far left, we're also working on designing some EPIC modules for them that bring various information together. And it's all based on the concepts that we had in that prior report. And it really helped the team there at Mayo also get on board with where they wanted to go. So each time there was a new lab or a new panel, they would approach it in this way. So we're continuing to work on those EPIC modules right now. So I'd like to touch on some of the more complex tools as well. So after years of being on parts of teams that included analysts working with biologists, so people doing AI or machine learning, working with either research clinicians or biologists, it became clear that even those collaborations were extremely valuable and necessary. The iterative process of the analysts working with the data, developing new machine learning techniques or tweaking approaches, and then discussing the results with the biologists and going back and rerunning or running new analyses, that process was just so slow compared to the questions that the biologists just really wanted to ask or what they wanted to verify. And so tools really need to be in their hands so that they could ask those next questions and run their analysis more fluidly. So this was really kind of the next mission then, was to develop tools that make it easier for the biologist or the research clinician to use really powerful data analysis techniques themselves. And so we continue to work in these collaborative teams, as I described, but when a new technique is developed by either a faculty member or our own researchers, we want to try to encapsulate that workflow and those approaches into a reusable workflow, so that then the data scientists, the analysts, can move on to developing the next new innovative approach, and the biologists then can use that recently established one themselves. So these are images, and then also that last diagram is from a project called Knowing. It was a platform for genetic data analysis, and this came out of the NIH Big Data Center of Excellence, where Saurabh Sinha was the PI and led the development of this platform. Our team was responsible for designing and implementing the visualization and the user experience for this platform. And what you see here is the images along the top. I know you can't see all the details, but they illustrate how the biologists can upload their data, step through a process to set up parameters for whichever analytical pipeline they want to use. Then they can execute the process on the cloud. And then when the results come back, they can study the results using these various interactive visual tools. This is one of the pipelines for gene set characterization using an approach called Drawer, which was developed by Charles Blatty, who is now a research scientist on our team. As with all of our tools, we provide a lot of flexibility for the researcher to compare data or to map results to data from different public databases, for example. We provide high-level views and the ability to access more detailed data and more detailed filters. And we provide multiple ways to look at the same results. And of course, the ability to download all the numeric data as well. In this panel, I just wanted to pull out one to point out that what you see here is that we also provide explanations, clear explanations of each option, so that the biologists can understand the analysis, how the analysis works, and then adjustments might affect the outcome. Which leads me to my next topic, which is explainability. And so explainability, as some of you may know, is an issue that really does hinder the use of some of the most advanced machine learning approaches, or deep learning approaches too. If the biologist does not understand the approach, it's difficult for them to really assess the results and also to understand maybe the variability or the pros and cons of the various techniques. So for this tool, we do provide diagrams of the techniques, so the biologist has at least a better mental model of the approach. And then we include reference publications, so the biologist can access more deep, in-depth explanations and the citations that they'll need when they need to describe the methodology in their own publications. In addition to a lot of the complex analytical pipelines in knowing, we also have a tool in there that we call the Spreadsheet Visualizer. And that helps researchers really just to get a sense of their data. It does a little bit of descriptive statistics, but it's really so that they can understand what's there before they start to execute their various more sophisticated analyses. So the tool provides a great amount of flexibility for the user to display and sort their data using all the different variables or metadata that they have in their various spreadsheets. We provide small graphs that show some of the distribution or shows the distribution of samples per phenotype, for example. And then the columns in each of these indicate a single sample or a single patient. And they're all lined up vertically to match each other across the visual displays. So you can include continuous or categorical data. And if you have spreadsheets with different data types, such as a spreadsheet of expression data and another maybe of metabolite data, you can display them together and the patients will all line up as long as the patient IDs are the same in each. And you can also calculate survival curves on the fly. So we still maintain the knowing platform at NCSA, and we're currently now expanding it, or we'll be soon expanding it to include new pipelines, and also working with our collaborators at NIH to integrate data relevant to some of the COVID-19 and flu pandemic analysis that we're doing with them. So the next tool I want to show you is, we call it Omics, and it's really focused just on microbiome data. So Mayo funded this work, and one of the objectives was to bring data from multiple studies into one tool and reprocess that data so that researchers could combine and kind of get value from a larger selection of data. The user can create then in the tool 2 cohorts to compare, and then run machine learning approaches. The one we have in here is a random forest approach that then identifies microbes that discriminate between the 2 cohorts. So often it will be a control cohort and a disease cohort, but not always. This panel, this particular view, shows a comparison of the composition of each cohort's microbiome at the phylum level, as well as a distribution of the samples in each of those cohorts, and it calculates a richness and evenness score. And then this is another way to look at the same data. This shows the relative abundances, but mapped to a phylogenetic tree so that the biologist can expand the branches however they'd like, compare different branches where they're interested in, and be able to see the relative abundance at each of those points. They get a better understanding then of how the cohorts compare and where they diverge. And then we also provide a way to display one patient's data in comparison to those 2 cohorts. In this case, the patient data is shown in green. We also display the results of the random forest analysis within the view. The relevant microbes, or OTUs, if you're familiar with microbiome data, are marked with red dots. I don't know if you can see them, it's pretty tiny. And then those are summed up as you go up the tree. And the researcher can easily see, for example, if many of the relevant OTUs are near each other on the same branch, or they can also see the quantity of significant OTUs at each level. So here you can see I've expanded it out all the way to the species level. And even down at the species level, you can do a comparison of relative abundances per cohort and with that patient, and see that distribution of even just that particular species or that OTU. one other detail I wanted to point out from this tool, which is also something we use in Knowing and try to put it in most of our tools. Often with results of analysis, there may be arbitrary thresholds that are chosen for a cutoff. So something like, displaying the top 100 genes or the top 10 OTUs. And so we like to came up with a way to provide, display the whole range of results. Here you see a graph showing the range of scores, and then a slider where the researcher can choose a more appropriate threshold based on the data. So if you move this, you can easily see how as you move the slider, the red dots below will appear or disappear depending on where you put the threshold. So this tool really puts some powerful machine learning approaches as well as some traditional descriptive analysis that they're familiar with using microbiome, and kind of an easy to use tool for the researcher to study their own data. And really, it's there to help them develop hypotheses that then they can go dig deeper about. And again, we provide some information on the complex analysis. And here we're planning to add another approach, the one that you see there on the left, developed here by Roqing Zhu in statistics that we work with a lot. We're hoping to integrate that approach into the tool as well. So another valuable project we did a few years ago for Mayo was the evaluation of their many genetic test workflows in their Mayo labs, and look at the tools that they use to evaluate, interpret, and then report patient results. So the goal that they had for us was to identify where the use of information design could help improve the efficiency of their workflows, as well as ensure accuracy of their interpretation of the genetic material. So you can see from this, very high-level diagram that one task that's relevant to each person in the workflow is that process of reviewing the quality and the coverage of the genomic sequencing. So one of the first projects they had us work on was that we've been working on since then is implementing this new component for this task. And the first release of it is now integrated into their clinical workflow. I'm not going to go into a lot of detail of this example since it's not publicly available, but you can see from these views that we continue to provide multiple ways to look at the data. We provide context and methods to navigate and move through the data. We use visual hierarchy to layer the information and differentiate between important primary and supportive information. And because it was tightly integrated with Mayo's cyber infrastructure, our team software engineers, led by Matt Berry, had to work very closely with multiple software teams at Mayo. So we're very, very proud of this tool, and we're going to be starting a second phase of this work next semester. Excuse me. The last tool I want to talk about is a current project we call Picture, which is led by Peter Groves on our team. And we started a collaboration with Steve Bopart to help analyze images created by his team's stain-free multiphotonic imaging technique. So we tried multiple deep learning and machine learning approaches. What I wanted to explain, if you're not familiar with image analysis, it's important to know that in some approaches, your results can be greatly improved if you have example images that are annotated well and then used to train the model to identify the features in the image. So for example, you would mark up, like in this example, draw boxes around the objects, such as a stop sign or a building, and label it for the AI system. But some images in the scientific and medical fields, such as Dr. Bopart's, are much more detailed and also include multiple layers. The complexity of labeling different cell types and structures in the image requires almost a pixel by pixel labeling and expertise to really determine the accurate features. So as we search for useful tools to help us with this part of the process, we discovered that other science domains face the same challenge, and they do not have adequate tools to do this at the level of complexity that we need. For example, our colleague in atmospheric science, Larry DiGirolamo, has a challenge to better distinguish clouds from snow in satellite images. And those are also multi-layered and massive. The Google Brain Team's AI model to identify diabetic retinopathy in images required that they pay many ophthalmologists to painstakingly annotate hundreds of images. And they also, we checked with them, they also did not have a tool for doing this effectively. So we have since been developing a tool to make the task easier and more accurate, and also to integrate machine learning into the workflow to aid in the complex and tedious process. Here are a few examples. Peter has explored ideas of replacing the intensity channel in an image and instead mapping the classification probability to that channel to help bring out certain features. We're using the multiphotonic images as our first scenario, and this is a mock-up of what's being implemented right now. We expect to have our prototype ready in January. And then we will soon be developing additional features to work with the satellite data for cloud detection. So Lisa Gatsky and Chad Olson on our team have also explored many different interactive techniques to help make the process more fluid and efficient. Not only is the annotation aspect critical, but also creating an environment to easily test the AI model on massive amounts of data and then be able to review the results against the ground truth images will be another component of this tool in the future. So I hope that all of these examples have demonstrated at least some of the key characteristics of how information design and user experience design could be used to address some of the challenges of using data effectively. I believe it's also a technique, as I described, to help develop innovative solutions. And through the design of mock-ups and prototypes, you can help people to develop a vision of what's possible and then have a clear goal of what to try to achieve. I think one of the reasons that NCSA has been a good environment for innovation is because of the many different sciences that are represented. And that makes it easier to kind of see some of the common challenges related to data and computation. And then, of course, we learn from each other and can pool our resources to make advancements. Just to give you one example, the movement and processing of massive amounts of image data will someday be very common in healthcare. And we can look to other projects and lessons learned, for example, from astronomy, and then leverage some of those technologies and infrastructures where possible. I continue to co-lead now the visual analytics team as a PI, and Matt Berry now manages the team and manages the projects. But in my more current role at NCSA, I've formed the Healthcare Innovation Program Office, and I'm focusing now on something that I've found to be critical, and that's identifying opportunities for collaborations and really trying to bring the right people together to address some of these hard problems. So I don't focus on visual analytics entirely, but along with Laura Martin, who's our program manager, I help other groups across NCSA expand their participation into the healthcare domain. These are the current program areas that we're working in. In addition to the software and data analysis that I've already touched on, one of the things I wanted to highlight here is also that we have a HIPAA compliant computational environment that we are standing up to support faculty research. So there have already been faculty coming to us who have patient data that really want to try to study that data and not have to de-identify it for various reasons. So I just want to make you aware of that, that if that's something you may need in the future to definitely contact us. And as you can see, there are many different areas where NCSA is engaged in biomedical research and clinical practice. And I'm happy to answer questions about any of these other things, either during the discussion time or you can contact me later about that. I'm also a member of the Illinois Mayo Alliance on the steering committee, and we're in the 10th year of that relationship. And this is an organization directed out of IHSI by Neil Cohen and managed by Deborah Miller. And there are many opportunities to engage with Mayo through this effort, including student fellowships and faculty seed grants. So I encourage you to check it out if you're interested at all. And I also want to mention that NCSA has a faculty fellows program. So if you're faculty and you want to collaborate with NCSA staff on your research, the next yearly round of proposals are due in March. So reflecting on all I've experienced, there are so many stories and so many lessons learned, but I think the most important that I've learned is understanding the value of really true good collaboration. And I've really been fortunate to experience great collaborators at various times throughout my career. And I've experienced a culture of collaboration at Mayo Clinic too, which has really had a strong influence on me. And I used to wonder, why was I so lucky to have many times in my career where I got to work with some great people that made an impact? But recently, I've really started to realize that it's not luck. It's just that there are so many extraordinary people around. So it's not as rare as I thought. There are just so many opportunities to come together with our various unique skills to tackle some of these problems or to create something new. So I want to end, I'm not going to read this, but I just wanted to share this as a part of a story from an excerpt from Salman Rushdie's children's book called Huron and the Sea of Stories. And I like how it kind of relates to the idea of collaboration and especially the last part where all these stories have been held in fluid form. They retain their ability to change, to become new versions of themselves and to join up with other stories and so become yet other stories. So I find that inspiring. And so I just want to thank you again for giving me the time to talk about all this. And I look forward to hopefully connecting with some of you in the future.
Very good. Thank you so much. That's wonderful. I think it just is a nice historical representation, but also, so much rich data and rich ways of viewing and visualizing that data and representing that data. And I think there's just, a lot of questions that maybe we can move into right now to probe further. I see there's one question from Zach Kircher. Again, kind of thanking you for covering such a impressive array of work, but also asking, you talked a lot about this collaborations and multiple institutions and what about the data sharing or the, kind of the intellectual property rights or the ownership of that data?
Yes, so well, we have, as you said, a lot of projects we work with various researchers, faculty, with Mayo Clinic, with NIH researchers as well. They share their data. We always set up a data use agreement, even though it's not always required by the institution we're working with. But there's always an IRB, whether the other institution or we hold it. And then we always have a data use agreement where we make agreements on how we're going to share that data. We've never had problems with that. I know with our Mayo collaborations, there's a master agreement in place for any research that happens with Mayo. The IP of anything new created as a joint part of that collaboration is a 50-50 split in terms of intellectual property. There have been some cases where maybe there was, an unusual circumstance, or sometimes we do service contracts with Mayo where we're developing a tool for them that they're paying for, and then in those cases they own more of the property.
I see. Okay.
But yeah, if you're interested mostly in the Mayo, Deborah Miller, as I mentioned, she has all the information about that. But what's great about the Mayo Alliance unit is that they're there to really help facilitate and get through all those barriers to really try to enable collaboration.
Mm-hmm. Great. So we can open this up. If anyone has questions, please unmute yourself and feel free to ask.
I cannot, while we're waiting, I can also make another comment about data sharing. And I mentioned that we have this HIPAA compliant environment we call Nightingale. We also have systems for data that doesn't need to be in a HIPAA compliant environment, but maybe it's still sensitive, so we want to be more secure with it. We're also trying to set up some rich data stores for people to access, especially people developing new analytical approaches where they really need to test on various types of data. And so our colleagues at NIH, for example, are very willing to allow us to take the data. They always will make their data publicly available at some point, but it's not always, as you know, easy to use, to find it and to have all the detail you really need about the data to be effective. And so we're going to work with them on a use case where we're going to take some of this data they have from their flu studies and make it available for faculty. And one of the reasons I feel that this data could be very valuable is because there are particular flu studies where they have many samples compared to other biological studies, in the 1,000 samples instead of just 15 or 20. And they are brought into the hospital there, and they're given the particular flu virus, and they take a lot of data before they give them the virus. So they have their DNA, they take all different types of blood samples, and then every day after that, and then every 5th day, and they take symptoms. So the same doctor is evaluating the symptoms for that entire time that they're in the hospital with that flu. And so it's a very unique set of data that it's that rich, and that the day prior, the prior, and then when they got that virus. Another set of data, we've been talking with Jennifer Erdley at Carle Hospital. She's very interested in providing data also for faculty and students to use, and we're trying to figure out the best way to do that, where it's wonderful to have some patient data along with some insurance data too, as kind of a unique type of data set. So we're also trying to find ways to help enable people to develop, to create new ideas, but not be burdened with that difficult task of finding quality data to start with.
Yeah, no, that's really important. I think all the students in Carle, Illinois will have a data science project in their last phase of their program. And I'm wondering if you have any suggestions or ideas or advice to them as to, where to come up with these particular ideas or topics or resources for those.
Well, I know Charles Blatty on our team is part of that effort to provide students with various tools and data to be able to get started. I think for those that really want to focus on that area, as they move forward, as opposed to getting enough experience so that they can participate in that. There are also opportunities at NCSA. We have an AI center led by L.U. Huerta, and they often will work with students or groups of students on particular projects over a semester. And so if there are med students that really want to take on an additional project or really dive deeper with some people that focus on not necessarily the health domain, but different analytical processes across many sciences, that could be a great opportunity as well.
I think I'll definitely use your quote you said earlier, it takes practice to take risks.
I think it does.
I love that. That's something we have to instill, right? And work at it.
Yeah, the first set of spin students that we had, I learned through that first one where it was just really, they were nervous about, once they picked a project, they were very afraid that they were picking the right one. And so I almost forced them to just pick something and then drop it later, so that it feels okay to drop it and start something else if it just didn't go the right way. But I do think it's almost like giving people permission to take chances, or it's okay to fail, can open up some doors.
Yes. Yeah. Other questions, please. The history you gave, starting from Mosaic, and it was wonderful to see. And I think that's something we take for granted, right? We're all using web browsers now as part of our daily life. What do you think the future is for the web browsers, the interface, the way that we kind of interface with this information, on a daily basis like this? Is there more innovation to come in that space?
That's funny you said, is there more innovation? Because I feel like at all those, interesting points in my career, I felt like, what new could possibly come later? And there's always something else that pops up. And related to information design, one of the things, just generally, not necessarily just in the medical space, but that I feel is kind of missing in our culture or in our younger education. Our kids, students spend a lot of time diving very deep into grammar and how to write, how to read, which is all very important, of course, but the amount of effort spent on teaching that compared to the amount of effort teaching people how to communicate with data, how to understand data, is just, there's no comparison. And to me, it feels that's going to become more and more important. People have to understand how to interpret things, how to be skeptical of data. I think a lot of times people that are not as experienced working in data just assume if it's data, or like as Edward Tufte found, if the diagram shows that, that must be real. And so I think it would be great to teach people so the next generation is a little bit more, really analyzes the data more before they decide whether they believe it or not.
Mm-hmm. Very good point. There's another question in the chat. Assad Ruff, if you wouldn't mind maybe asking that, or I can read it directly, but
you can see it.
With these made-to-order solutions that are made for specific use cases, how much thought do you give on how applicable the solutions might be for other cases?
Yeah, that's a good question. I didn't mention at all that our philosophy, and even not just in our group, but the software directorate at NCSA, is really, we try to make everything we do as generalizable as possible. So even if, like for example, in the image annotation software that we're doing, if we only focus on one scenario, we could probably figure it out a little more quickly and implement it more quickly. But we're trying to figure out how does this relate to many other scenarios? How would we change things? How do we make it very modular so that we can build on it? Because we're always thinking about how to expand to other communities or how to leverage anything else we have prior. So like with the Knowing platform, we try to leverage as many pieces as we can, so we're not wasting time, rebuilding something. And really, we're always trying to make it as useful broadly as we can.
Mm-hmm. Yeah, that's an interesting point too. One of the questions I had put in the chat was, how do you account for people's, the psychology of that individual, right? The viewer. Their perception, any biases they may have, right? They're always going to approach data and visualization from their own lens.
Right.
How do you address that variability, right?
Yeah, I think we try to, you try to engage with as many different kinds of people and different perspectives as possible, just so that you can kind of see how they would want to use something or how they might interpret it. And really, I think it comes back to that flexibility that we try to put in tools. It's like we try to represent it in multiple ways. We try to create different views or different ways to move through it to try to accommodate, different types of personalities, different types of tasks. You might be the same person, but coming to the tool for a completely different reason. You need to get something fast. You need to answer a question fast. You want to go through it a completely different way. So we try to accommodate as much as we can.
Mm-hmm. Yep.
And that's why I should say one of the reasons I was excited to talk with, future med students and people in this field. It's what I've found, people have ideas of maybe how they, if I was a clinician, a clinician might be how they would use a tool, maybe what they would want in a tool. They don't always know the best way, and same with the researcher, to talk with the designer to kind of share those thoughts. And I thought providing this kind of presentation might give people some insight to what is the designer trying to figure out. So they don't necessarily want to know, how do you want to do this? They want to know, what are you thinking? What are you trying to do? What do you want to do next? What are you trying to answer? And then let us figure out, show you ways. And often we'll come up with multiple ideas of how to accomplish whatever question they're trying to ask, and go through an iterative process. It's definitely iterative.
Mm-hmm. Mm-hmm. Okay. I see Kenny had a question, so it's more technical, but what data visualization library have you found to be easiest to use for web app development? And is there a programming language that you tend to find more common or useful?
Yes, and I would be nervous answering that since Matt Berry's not on the call, unless he happens to be on the call and he can chime in. I would have to have him answer that for you
Mm-hmm.
because I don't want to mislead people if I say something that's old that they no longer use. I don't want to give the wrong impression, but they definitely have preferences. They also, the team has worked in different technologies. Some of the work we do matched pretty well with what the Mayo developers do as well. We might have tweaked a
Mm-hmm.
few ways we do things to match up with them, but we try, especially with some of the research and prototype tools we develop, we also develop them thinking down the road to, okay, if this becomes something used in the clinic or needs to be integrated, we don't want it to be a complete rewrite and throwaway code. So we're always developing with the intention that it's going to continue moving down that development path and become more solid to kind of make that smooth. So if you're interested in that area, that's definitely something our team has a lot of experience with and can really talk about that continuum and then what technologies have been valuable in that continuum.
Mm-hmm. Okay. Yeah, Kenny indicates he'll reach out to you after this. It does have a follow-up question too, is that, for students that are looking to be exposed to all the different types of data visualization methods and advantages and disadvantages, is there a resource, right, that you would recommend for that comparison?
Yeah, that's good. Kenny, I'm going to send you some other links too, because off the top of my head, I can't think that there are various websites that you can go to really look at a variety of different techniques that people have used. I also, there's certain places that tend to create, like the New York Times tends to make a lot of interactive graphics that follow a lot of the same kinds of principles of visual hierarchy and not always, but a lot of times those I've looked at those and shown those as kind of inspirational solutions to ideas. Edward Tufte's books, of course, if you're not familiar, it's great just to look at examples even, and of course read some more information about ways to approach things or tips. So those are all good resources.
So you had mentioned scalability in your presentation, and are a lot of the I guess the methods, the structure for this type of data design and visualization, does that scale? So I'm thinking from the medical perspective, we talk about kind of these emergencies. You have a single patient emergency, and there's a lot of data that needs to be exchanged and visualized. But then that also can scale to, public health and to global pandemics, right? And so is that visualization scalable as well from like patient to a global scale?
Thinking back to some of the like situational awareness kinds of scenarios in those variety of tools, I think there are definitely different tasks that you're trying to do, even different kinds of data that you're trying to bring together. But I think if you build off of some of the same concepts, the same metaphors, it helps somebody keep that context. So even if you maybe are using some sort of tool that's more of an overview of your entire practice, for example, where there's a way that you can get a sense of various pieces and then a way to look at certain kinds of information. If you use some of those metaphors again, or those same ways of organizing information, but in a broader context, it helps that transition, across that scale.
Mm-hmm.
Yeah, it really depends because some challenges just have their own unique problems that need to be addressed.
Yeah, very good. Other questions that the audience may have?
I have one. Can you hear me?
Yes.
Great. You've been talking so much. I mean, it's impressive work, Colleen. Every slide that you presented is probably a couple of years of work. Very.
Helps
and he
to
went.
be old.
No, it's just an incredible amount of work. Very impressive. At Geese, we're trying an initiative I'm trying to lead, something that I call data cases, which brings real data closer to students. So we're going to use them in classes, in courses, and that kind of hopefully bridges the gap between training in the business school to actual using of data outside. We're going to pilot test it with big data. Hopefully we're going to get soon from the Cook County Assessor's Office. And we really want to engage companies in that as well, because the idea is that they need that. We have some statistics that suggest 80% of companies only do, exploratory data analysis with the data they have. We think we can help them to do a lot more with our undergraduate and graduate population, as well as faculty. So I think could be a win-win scenario. The question I have for you is, do you have experience convincing these kinds of data partners or establishing these kinds of data channels with partners outside of the Mayo Clinic, which seems to be a very, very involved and very useful partner?
Mm-hmm. Yeah, I can comment on NCSA more in general. We do have an industry program. I'm not sure if you're familiar with that, where we have industry partners that both use computation primarily, but then also engage in various other projects. And so I think I would love to get you in touch with Brendan McGinty, who runs our industry program, and also Eliu Huerta, who runs our AI Center, because I think that could be a great kind of collaboration with your college to help facilitate, even to provide not only tools but computational resources, and somehow could be even some of our industry partners may be interested in providing some of those data resources and challenges, like you said.
Fantastic. I'll follow up with you by email afterwards. Thank you, Colleen. Wonderful talk.
Thank you.
Okay, any other comments or questions, thoughts, ideas? I think another question in the chat there. Are there traits or characteristics you find important in clinical partners in terms of making them easy to work with?
Yeah, I would say not just clinical partners, but really anybody. The most successful projects we've had are with people that are just really passionate. They're excited to try something new. They're willing to take some of those chances or be open to ideas, but then also give good, concrete, give their opinions, so it's not just they're open to everything, but it's really critical. They dive in deep, and that's what we need. Is we need someone to be able to say, well, this is where a problem could occur. If I'm going to meet with a patient and I don't know this, we want to know all of those potential problems. And so it's somebody that is very open and enthusiastic, but then also dives in deep. And so I think, and also with a lot of our projects, our collaborations with Mayo, the ones that work out the best, we really do work closely, like, at least once, a week, with our software projects, multiple times in a week. With some of the clinical researchers, the ideal ones, we usually at least meet once a month. At least those are the ones that seem to be the strongest. And and really, this is kind of a weird thing, but I've sort of found over time that the groups, not just clients or clinicians, but even teams that I work with, the ones that we laugh the most seem to be the ones that we just really engage on things. And I think maybe it has something to do with you just are at a certain comfort level that you can just really work well together. So I've kind of learned that that's a little bit of a sign too. It's just really building a relationship with people.
Yeah. No, that's really interesting. You have to have relationships with the people before you have the relationships with the data.
Yeah, that's good. I'm going to put that in my next set of quotes.
Well, thank you so much. And again, on behalf of everyone here, thank you for your presentation and the historical perspective and insight. I think this is something for us all to think about, and we're all going to encounter so much data in our lives that we are going to need to think about and use tools like this to make sense of it. So thanks again. I want to wish everybody happy holidays and a little break. We're going to reconvene in January with as these start up again, but wish everyone well.